Hi everyone, welcome back to my channel. Today I'm going to take you through the Digestion and Absorption section for AQA A Level Biology. Also, I'll be going through some exam style questions and their mark schemes. And as always, in the comments section I'll be putting timestamps so that you can skip to the relevant section that you want to revise from if you don't wish to watch the whole video. Now, this is quite a short topic as it kind of just builds on the knowledge of enzyme action from the biological molecules unit. If you haven't already watched that video, go and watch it now. This topic kind of just builds on that, just talking about the digestion parts of enzymes. Let's get started. So here is an image that I've stolen from the internet of the basic human digestive system. So here we've got the salivary glands, which is important in producing the enzyme amylase, the esophagus, which is the food pipe, the stomach, the pancreas, the liver, the small intestine or the ileum, the large intestine and the rectum, which is the place where feces exit the body. So the first enzyme that I'm going to talk to you about is amylase. Amylase you probably have heard of before. It's produced in the salivary glands or the pancreas. So the salivary glands are these parts here near the beginning of the digestive system. If you feel the top of your neck, you may be able to feel your salivary glands. Now, amylase is also produced in the pancreas. Just get my pen out now. Pancreas is this little organ here which kind of sits just below the stomach on this diagram. Now, pancreatic amylase is secreted into the small intestine. Now, the job or the function of amylase is to hydrolyze or break down starch, which is a carbohydrate or a polysaccharide, into maltose, which is a disaccharide. This leads me on to explain the role of membrane-bound disaccharides. Membrane-bound disaccharides, as the name suggests, are present in the membrane of the small intestine or the membrane of the ileum. The job of membrane-bound disaccharides is to hydrolyze disaccharides, for example, maltose or lactose or sucrose, to monosaccharides, for example, glucose. Now, membrane-bound disaccharides can be, for example, or disaccharidases, I should have put here, not just disaccharides, disaccharidases, just a typo. So these can be maltase, sucrase and lactase. And they function in the small intestine, which is this squiggly folded part here. So the next enzyme you need to know about is lipase. As the name suggests, lipase hydrolyzes or breaks down lipids to monoglycerides, so glycerol, and fatty acids. Lipase is present in the small intestine. So you need to know about some adaptations that lipase has to make sure that it functions efficiently and quickly. The liver, which is an important organ in the digestive system, makes these things called bile salts. What these bile salts do is they emulsify the lipids. This gives them a large surface area, which means that they're easily accessible and easily hydrolyzed by lipases. Now, the products of lipid hydrolysis, so monoglycerides and fatty acids, can remain associated with these bile salts to form structures called micelles. And what these micelles do is they travel to the ileum, which is part of the small intestine, and are broken down when they come into contact with the epithelium. The epithelium is a collection of cells which line the lining of the ileum. The products of this broken down process can then diffuse straight into the epithelium, which is a good adaptation as the absorption of the product is faster. So now you need to know about proteases. Proteases, as the name suggests, hydrolyze or break down proteins or polypeptides into amino acids. Now, there are three types of proteases that you need to know about: endopeptidases, exopeptidases, and membrane-bound dipeptidases. So let's talk about endopeptidases. Endopeptidases are, for example, trypsin, chymotrypsin, and pepsin. You may recall from GCSE that pepsin is the protease present in the stomach, as it can survive very acidic pHs. So endopeptidases hydrolyze peptide bonds, which are the bonds that connect amino acids, in the middle region of the polypeptide chain. So here we have a relatively short polypeptide chain here. So endopeptidases, as you can see, hydrolyze peptide bonds in the middle region, indicated here by the arrow. So as you can see, this is kind of in the middle, it's not at the ends. Which leads me on to exopeptidases. The difference between endo and exopeptidases is that exopeptidases hydrolyze peptide bonds on the terminal amino acids. So here we have our short polypeptide chain again, and exopeptidases can hydrolyze peptide bonds on the terminal amino acids. So here we have our one of the terminal amino acids. So exopeptidases can act here. Now, the way I used to remember this is that it is the opposite way round to what you think it is. So you would think that endopeptidases starts with the word end, so you would have thought that endopeptidases hydrolyze the terminal or the end amino acids, but it's the other way around. Endo hydrolyzes the middle peptide bonds, and exopeptidases hydrolyzes the terminal or the end amino acid peptide bonds. So lastly, we're going to talk about membrane-bound dipeptidases. So dipeptidases, as the name suggests, hydrolyzes dipeptides, which are two amino acids condensated together into two amino acids. So as you can see, we have two amino acids joined together by a peptide bond, and membrane-bound dipeptidases present in the lining of the ileum or the small intestine hydrolyze the peptide bond between these two amino acids, as indicated by the arrow. So the last piece of content that you need to know is how amino acids and monosaccharides like glucose are absorbed into the blood from the small intestine. This is through the process of cotransport and is basically the same process as I talked about in the absorption of glucose in my transport across cell membranes video, which you should check out if you haven't watched already. So this is how it works. It works through the process of cotransport. Cotransport is when two molecules are transported together, one down its concentration gradient and one against its concentration gradient. So this particular method uses sodium ions, Na+, along with the amino acid or the monosaccharide, so glucose. So the first step in the process is sodium ions and the amino acid or monosaccharide are co-transported from the lumen, which is the space inside the ileum, into the epithelium, which are the cells that line the ileum. So they move into the epithelium by co-transport through channel proteins. Sodium going down its concentration and the concentration gradient even, and the amino acid or monosaccharide going against its concentration gradient. Therefore, this requires ATP hydrolysis to work. So the next step is that sodium ions in the epithelium are actively transported from the epithelium into the blood, and at the same time, potassium ions are actively transported from the blood into the epithelium. This is called the sodium-potassium pump, and this ensures that a favourable concentration gradient of sodium is maintained from the lumen to the epithelium, which ensures that the substrate, so the amino acid or the monosaccharide, can be efficiently transported into the blood. So therefore, the last step is that the substrate, so the amino acid or the monosaccharide, is transported into the blood from the epithelium through the process of facilitated diffusion. Now that is it for the content. Now I'm going to get on to some exam style questions. So here is the first question. I'll just get my highlighter out. So the diagram represents parts of the human digestive system. The organs are labelled A to F. So here we have a diagram of a pretty simplified digestive system. Now, here's the question. Give the letter of the organ that produces amylase. So I said before that amylase is produced both in the salivary glands and the pancreas. Now, this diagram obviously doesn't show the salivary glands, so we want to look for the pancreas. Now, as we noted before, the pancreas sits kind of below in the stomach. So we can say that this here is the pancreas, this structure here. So C is the answer. So let's look at the next part. Give the letter of the organ that produces maltase. Maltase, as I mentioned before, is a membrane-bound disaccharidase. So this means that it is present in the lining of the small intestine or the ileum. And the ileum here is represented not by tight folds, but just this single tube here. So we can say that the answer is E. So if we look at the mark scheme, C and E both get those marks. Now it says here, ignore the name of the organ. So you don't get penalised if you put the letter and the organ, but if you just put the name of the organ, you don't get the mark. So if we look at the next question, this has more to do with enzyme action, as I talked about in my proteins and enzymes video. So maltose is hydrolyzed by the enzyme maltase. Explain why maltase catalyzes only this reaction. As this is an explain question, you need to write down why something occurs. So you need to write down why maltase catalyzes only this reaction. So this question is asking us about enzyme specificity. So this is what I've put. Maltase's active site, the key term here is active site. You can't just put maltase. You need to refer to the active site. Maltase's active site has a specific tertiary structure or a specific shape. So only maltose can bind to the active site to form an enzyme-substrate complex. Now, an examiner likes it when you use the term enzyme-substrate complex, so always try to use that when you're talking about enzymes or enzyme specificity. So let's look at the mark scheme. Active site, so as this is underlined, you need to write the term active site, otherwise you don't get the marks. So active site of the enzyme has a specific shape or a tertiary structure, or you can write active site is complementary to a substrate or maltose. Now here it says reject active site on the substrate, as the substrate itself doesn't have an active site. The active site is on the enzyme. And as it says reject, if you write the active site on the substrate, you don't get any marks at all for the question, even if you wrote the correct answers for mark point 2 and mark point 3. Must have the idea of shape or tertiary structure. Assume it means maltase. So if the examiner thinks what you've written as it refers to something else, you don't get marks. You need to make it really clear that it refers to maltase. Accept specific 3D active site, as the tertiary structure is 3D. But you have to reject has the same shape, as enzymes and substrates do not have the same shape. If they had the exact same shape, they couldn't bind together. So the second marking point, only maltose can bind or fit. It says accept substrate for maltose, but I recommend that you use the term maltose as it's more clear in your answer. And the last marking point is to form an enzyme substrate complex, and they accept ES complex if you can't be bothered to write enzyme substrate complex. It all happens to all of us. Right, next question, 4-mark question. Describe how proteins are digested in the human gut. As this is a describe question, you don't need to explain why something is happening, you just need to write what is happening. So this is what I've put. Proteins are broken down through the hydrolysis of peptide bonds, which kind of gives your background into the digestion process of proteins. And now you need to delve more specifically into the types of proteases. So endopeptidases hydrolyze polypeptides into smaller peptide chains by breaking the middle peptide bonds. Exopeptidases remove amino acids at the termini, and dipeptidases hydrolyze dipeptides into 2 amino acids. So let's look at the mark scheme. Hydrolysis of peptide bonds, which we wrote. Endopeptidases break polypeptides into smaller peptide chains, which we wrote. Exopeptidases remove terminal amino acids, or dipeptidases hydrolyze or break down dipeptides into amino acids. So we will get 4 marks. This is the last main question that we're going to look at, which is more of a problem-solving question. So let's read it. A student investigated the effect of chewing on the digestion of starch in cooked wheat. He devised a laboratory model of starch digestion. We can highlight this as well, effect of chewing, in the human gut. This is the method he used. Volunteers chewed cooked wheat for a set time. The wheat had been cooked in boiling water. This chewed wheat was mixed with water, hydrochloric acid, and a protein digesting enzyme and left at 37 degrees for 30 minutes. A buffer was then added to bring the pH to 6 and pancreatic amylase was added. The mixture was then left at 37 degrees for 120 minutes. Samples of the mixture were removed at 0, 10, 20, 40, 60 and 120 minutes, and the concentration of reducing sugar in each sample was measured. Control experiments were carried out using cooked wheat that had been chopped up in a blender, not chewed. Let's look at the first part of the question. What reducing sugar or sugars would you expect to be produced during chewing? Give a reason for your answer. So as the question says, pancreatic amylase is used, we can put maltose because, as I said earlier, in the salivary glands, which are part of the chewing process, amylase is produced, and amylase, salivary amylase, breaks down maltose, mean starch, into maltose. So maltose is present. So let's look at the mark scheme. So first marking point is you mentioned maltose, and the second marking point is that salivary amylase breaks down starch. So this is the next part of the question, pretty simple, just a 1 marker. In this model of digestion in the human gut, what other enzyme is required for the complete digestion of starch? Now, as starch has already been digested to maltose, we now need to digest the maltose into glucose. And the enzyme that does this is, as I have mentioned before, maltase. So maltase is the correct answer. Let's look at the next question, part of the question even. What was the purpose of step 2, in which samples were mixed with water, hydrochloric acid and pepsin? Now you can probably notice that water, hydrochloric acid and pepsin are all characteristics of the stomach. And as this experiment was carried out externally, we can write that the purpose of this was to mimic the effects of the stomach so as to get a more accurate result. However, you do not need to provide an explanation for your answer, as it is only a 1 mark question. But the mark scheme says to mimic or reproduce the effect of the stomach. So let's look at part D. In the control experiments, cooked wheat was chopped up to copy the effect of chewing. Suggest a more appropriate control experiment. Explain your suggestion. Now, as this is a suggest question, this often requires your own ideas instead of knowledge from the syllabus or the specification. So this is what I have suggested. I've suggested so that you add boiled saliva so that amylase is denatured due to the high temperature, but everything else stays the same. So you're changing the nature of the amylase, but everything else remains the same, providing an effective control. So if we look at the mark scheme, add boiled saliva. And the second marking point, everything the same as experiment, but salivary amylase is denatured. Now, I want to point out that the purpose of a control experiment is to prove that a particular process is changing the results and nothing else is controlling it. So now we're going to get on to the final part of the question. The figure below shows the students' results. So we have this nice looking graph here, one line for chewed cooked wheat and one line for chopped cooked wheat. Here's the question. Explain what these results suggest about the effect of chewing on the digestion of starch in wheat. Now the first point that I want to point out is, as you can see by the chewed cooked wheat line, it doesn't start at 0. This suggests that some starch has already been digested in the chewing process before the experiment started. Also, the gradient is faster, so we can say that chewed starch also has faster digestion rate, as the mean concentration of reducing sugars increases more rapidly than chopped cooked wheat. However, at the end of the experiment, so 120 minutes, the mean concentration of reducing sugars reaches the same. So we can put, however, the amount of digestion with chewed and chopped starch reaches the same point at the end of the experiment. Now let's look at the mark scheme. So mark point 1, some starch has already been digested when chewing or in the mouth. Faster digestion of chewed starch. And the last marking point 3, the same amount of digestion without chewing at the end. So we would get all 3 marks for this question. Here it says accept use of values from the graph. So it accepts examples of the values. So for this first marking point, you can put the concentration of chewed cooked wheat at the start of the experiment is around 60 or 70, I think. So you can use values, they accept that as well. Right, that is all I want to say. Thanks for watching. If you have any questions, please comment below and I'll see you in the next video.